Welcome back to Lucid Science. This week, we'll be looking at the mechanisms and behavioral functions of structural coloration in cephalopods by Mathker et al. First, let's go over some terms. Cephalopods are a type of mollusk characterized by bilateral body symmetry, a prominent head, and a set of arms or tentacles, such as octopuses, squids, and nautiluses. Chromatophores are pigment-containing or reflective cells that are found in a variety of animals, including amphibians, fish, reptiles, crustaceans, and cephalopods. Iridophores are pigment cells that reflect light using plates. When illuminated, they generate iridescent colors because of the diffraction of light within the stacked plates. Lucifores are flattened, elongated reflective cells found in the skin of many shallow water octopods that often reflect white light. Alright, I think we're ready, so let's get into it. Animals can be extraordinarily colorful. While in some animals, these colors help to camouflage their owners, in others they are used for signaling. And in some, like cephalopods, they can do both simultaneously. Cephalopods display an incredible range of body patterns, despite their apparent colorblindness. These patterns are displayed by thousands of chromatophores and structural reflectors like iridophores and lucifores working together. The ability to change the physical structure of chromatophores and iridophores is what allows these animals to produce such a wide variety of visual effects. A chromatophore consists of a pigment-containing sac that has dozens of muscles encircling it on all sides. The visual patterns produced by these cells can be seen in Figure 1, where A through D show the different patterns that cephalopods can produce, and E through I show the microscopic structure of the cells that produce them. In H, chromatophores are the large red cells, and iridophores are the small blue-green cells. By selectively expanding and retracting distinct groups of chromatophores, cephalopods can produce an array of patterns such as bands, stripes, and spots. We will focus on three specific patterning effects: iridescence, reflectance or mirroring, and camouflage. First, let's talk about iridescence. Iridophore cells are named for their ability to produce iridescent patterns by reflecting and dispersing light. Iridophores are generally located beneath pigmented chromatophores. They are colorless cells smaller than 1 mm that contain stacks of thin plates that reflect light by bouncing it between these plates. The mechanism of reflectance is the same as that of colored soap bubbles. If the soap film is very thin, shorter wavelengths are reflected, displaying blue light. If it is thicker, longer wavelengths such as yellow and red are reflected. This is the same for the thickness of the plates. Another way of changing iridescence is by expanding and retracting the overlying chromatophores. Chromatophores are generally located in a distinct layer above the iridophores, and by expanding, they can either change the reflected spectrum of the iridescence, create contrast against which iridescence is viewed, or block it altogether. This can be seen in Figure 3. For human observers, creating color is probably the most notable aspect of cephalopod iridophores for our eyes. However, when viewed at certain angles, cephalopod iridophores also polarize light. This is important because it has been found that the polarized aspect of iridescence may have communication functions. This has been termed a hidden communication channel because cephalopod predators are believed to not be polarization sensitive and would therefore not be able to perceive this sort of visual information. The final appearance of the animal is frequently the result of the interactions between iridophores and chromatophores. In fact, Mathker and Hanlon showed evidence that three color classes of pigments, yellow, red, and brown, combined with a single type of reflective cell, can produce colors that encompass the entire visual spectrum. This simple pairing is what enables the extreme visual diversity used by cephalopods for camouflage and signaling. Cephalopods have very striking silvery iridescence around their eyes. When viewed closely, the skin shows some variation in color. Some parts reflect red, others green and blue. When viewed from a distance, however, the various colored parts merge together, and the net effect is a mirror-like appearance. This can be seen in A and B of Figure 5, where A is from a distance and B is on a microscopic scale. The orientation of the reflective plates is such that the light reflected from almost any angle equals the intensity of the background light against which the animal is viewed. This arrangement would give close to a perfect camouflage, because it allows the animal to completely disappear into its background. In addition to iridophores, cuttlefish and octopus have another structural reflector cell that creates bright white patterns, shapes, and spots. These reflectors are called lucifores, or white cells. Lucifores reflect the ambient wavelengths of light, meaning that they look white in white light, red in red light, and blue in blue light, and are found in high intensity in specific areas that can be bright white, depending on the body patterning that is shown. This can be seen in Figure 6, where the white stripes are produced by lucifores. Okay, let's wrap it up. Studying animal structure coloration is mesmerizing, not only because of its sheer beauty, but also because of what we can learn from these structures in our efforts to produce electronic visual displays, as well as various kinds of paintings and coatings. So that's how cephalopods display such incredible patterns to blend into their environment and communicate with one another. Find us on social media at Conservation Made Simple, and I'll see you in 2 weeks for more lucid science.